Good morning. Happy Tuesday. Hope you're all doing okay this morning. So today, I don't have a whole lot of new stuff for you. We're going to go over the last bit of the organic chem stuff, which is reactions. So last time we talked about these functional groups, and one of the reasons we talk about those is different functional groups are going to behave differently in reactions. And so we're not going to go through all of them. If you were in an organic chemistry class, you would look at the different reactivities of different types of groups, and then we will you'd go through and look at those different reactions. So for us today, we're just going to look at one example. We're going to look at some of these carbonyl functional groups. So the ketones, the carboxylic acids, esters, amides, aldehydes. We're actually going to focus just on these last three, the carboxylic acids, the esters, and the esters and the amides. And we're going to look at one class of their reactions. So there are some quiz questions about this, the last three or four. So if you tried to do the quiz and those didn't really make sense, hopefully this will help you out. So good morning, everybody. Good morning, Alyssa. Good morning, Daisy. Let's look at some of these reactions. Morning, Jennifer. All right. So the specific reactions that we're going to look at they're basically in the same category. One's kind of like the forward reaction, one's the backward reaction. And the reason that we selected these particular reactions is because they're really biologically important. So when we go on next week and start talking about carbohydrates and proteins and lipids and things, we're going to come back to these reactions. These are the reactions that are going to be involved in those. So the reactions we're looking at are, we'll look at the terminology first, and then we'll look at some examples of what that looks like in the molecules. So first we've got hydrolysis. And hydrolysis is any reaction where you add water, that's the hydro part, and then you're going to break up a molecule into two. So that's the lysis part. Remember we talked about lysis in the context of cells. Lysis is like a cell breaking, or bursting. In the context of molecules, hydrolysis then is you add water, and the H goes. You'll see the reactions when we look at the molecules. But you add water, and then the molecule breaks into two parts. That's hydrolysis. The opposite of that reaction has a couple different names. It's either known as a condensation or sometimes dehydration. And that is when you remove or lose water and connect two molecules. Oops, I spell that right. All right, so we got hydrolysis, add water and break up the molecule into two, and then condensation or dehydration, water gets removed and two molecules get connected. Okay, let's look at some of those some examples here of those reactions. So we'll first look at condensation and dehydration reactions. So there are various ways to do this. We'll start with a very general sense where we kind of look at all of it like a Lewis structure where everything's drawn out in great detail, and then we'll look at some examples and get back to our line structures or skeletal structures. So in a condensation reaction, and kind of a classic one, you would have some carboxylic acid, which is like that. And we're going to use this R again as kind of a wild card of like could be whatever, any kind of carbon hydrogen stuff. And then we're going to react that with a couple different possible things, but our first example is going to be an alcohol. So we have a carboxylic acid, and we're reacting that with an alcohol. And I'm actually going to draw the alcohol backwards just so we can kind of see it. Not really backwards, but just this way. All right, so here's our carboxylic acid, and here's our alcohol. And actually, now that I think about it, let's make the alcohol a different color just so we can track things better. All right, so when these two react together, what's going to happen is, remember the definition of the condensation up here is that we're removing water and connecting two molecules. So the water that gets removed is here. It's the OH from the carboxylic acid and one of the hydrogens from the alcohol, and that makes H2O. Get it? Two H's and an O. And so that can leave, and then since the other bonds have to stay connected appropriately, then the carbon and the carbon from the carboxylic acid and the oxygen from the alcohol are going to end up being attached. All right, so let's draw the result of that, and then you can see how this works. So the rest of the carboxylic acid stays intact, but now it's no longer connected to this OH because that has left and become water, which we'll draw as a byproduct in a second. Instead, we have the O and the R prime, or this thing, from the alcohol. So the byproduct here is water, and let's be really careful about where that water comes from. It's one H that was on the alcohol, and then the O and the other H from the carboxylic acid. All right. Now you'll notice that this molecule here is now a different kind of functional group. Without looking back at the previous list, can anybody name what kind of functional group we have now? Like what kind of molecule we've made? Yes, exactly right. That's an ester. So we now have an ester. All right, so we were able to condense a carboxylic acid with an alcohol and come up with, or make, or form an ester. So this particular type of condensation might also be known as esterification, like we're making an ester, we're forming an ester. All right. Now let's look at a different kind of condensation reaction. Well, we should say a very similar kind of condensation reaction. We're going to keep that same carboxylic acid. And this time, let's react that with an amide, or I'm sorry, with an amine. So instead of an alcohol, we've got an amine. So just nitrogen connected to some carbon stuff. And this time, see if you can draw the product, or the products like we did before. So follow this exact same procedure, lose water, connect what's left, and see if you can draw the product and find out what that is. And if you want to guess that functional group, what that functional group is once you draw it, um, you can do that too. All right, hopefully you had a chance to play with that a little bit. So, um, yep, and you're correct. That's an amide is the product. So one of the important things to note here is we're doing the exact same thing. We're forming water, which is leaving, and then connecting what's left. So there's water, right? H2O. And then that means that the nitrogen is going to end up connecting to the carbon. Now notice what is a little different about the ester we made before is the hydrogen is still there. That's not part of this reaction. And one really important thing about organic reactions that sometimes is easy to forget is that the reaction is only going to affect those pieces or those atoms that are directly affected by that. There's sort of a temptation to make it mess with all other stuff in the molecule, but you got to resist that. Leave the rest of the molecule alone and only change what it is that the reaction specifically changes. So our product from that will be the R and the carbonyl from the carboxylic acid. And then from the amine, we're now connected to the nitrogen. And that still has its one hydrogen here. That hasn't changed. We can draw it with a bond or we can just condense it onto the N. And then coming off from there is the other R group. All right. So that's that. And then our byproduct is the same as before. We have an H. Sorry, you can't see that. We have an H from the amine, and then we have the OH from the carboxylic acid. All right, so that's condensation reactions. Now let's look at some bigger, maybe, molecules. Some more actual examples of molecules instead of just R's, and see if you can draw some of these condensation products. You know, amide, it would be a amidification. I have not heard that term actually, and I don't know why, because it does make sense. If we have esterification, we should have amidification too. Maybe that exists. I don't know. One possible reason for that, or possible difference here, is that this is an amide. Also, we mentioned that this is sometimes known as a peptide. So when this thing forms, when we form that amide, we're often doing this in the context of amino acids and hooking them together that way. Yeah, so good question. So one thing, whoops. So the question is, you know, if we're being asked to write a reaction for this, should we also write the water? And the answer is yes and no, or kind of it doesn't matter. You are correct that in a balanced chemical reaction, we need to account for all the atoms. So if water is coming off of the molecule, then that should be a product. And it doesn't have to be plus HOH. I just did that so you could see very specifically with the colors where those H's and OH's were coming from. You could just write plus H2O. But you'll sometimes find that that's left off. In organic chemistry, we tend to focus on the molecules that are changing, like the major things that are connecting or disconnecting, and often the little byproducts are assumed but not explicitly written. So if you're looking at a book or something and you're looking at condensation reactions and you notice the water isn't shown, sometimes it's just assumed. But if you're really being careful and trying to draw everything that's happening in that reaction, you should also draw that water as one of the products. All right, so here's a couple. Here's some bigger molecules to try out. The other thing that you'll notice in organic reactions is rather than the way we drew inorganic reactions, where it's like this plus this and then the arrow, often the main molecule is drawn on the left, and then the reagents or the things that are being added are added over the arrow like this. So this would be like saying, so this acid is reacting with this alcohol, ethanol, and then below the arrow, there may be other kind of co-reactants that just sort of help the reaction along. Like in this case, this reaction only really happens in the presence of a catalyst, particularly an acid catalyst. So you might see something like H3O plus here, or sometimes just H plus or things like that. But we generally focus on, all right, what are the major things that are happening? We have a carboxylic acid reacting with an alcohol, and that's going to be a condensation reaction. So we want to draw the condensation product here. So see if you can draw that, and I'll give you a couple other examples also. All right, so start with those two. See if you can draw those products. Now this looks a lot different from when we had it all drawn out very carefully atom by atom before, but it's the same process, and see if you can figure out what's happening in this case and then draw those products. If you're totally lost or um, you're not sure about how to draw something, I wish I could see what you're drawing, but at least if you can try to add something in the chat about like, well, what do I do with this atom? Yes, you will have to remove H2O just like before. That's correct. I think so. The thing that's going to be a little bit trickier here is just making sure you know which H, H, and O to remove. No, we're not going to include the acid at the bottom of the arrow. That's a catalyst, so that's not used up in the reaction, and we can just kind of ignore it. Yeah. Okay, let's take a look at these. So what I was saying about picking the right hydrogens, it's mostly just about understanding the idea of the functional groups. So the this molecule is full of hydrogens all over the place, right? It's got 2 oxygens and so forth. This molecule is also full of hydrogens. You can't just pull any hydrogen you want to make the water. You specifically need the OH from the carboxylic acid and the H from the other functional group, either the alcohol or the amine. So you can't just go pulling hydrogens off of carbons and connecting that way. And similarly, we can't mess with the carbonyl oxygen because we know that stays there in the process. So the hydrogen that we're taking is going to be for the water is going to we're going to take the OH from the carboxylic acid, and we're going to take the H from the ethanol. And you don't have to draw this circle thing. This is just the way that I think about it. This one plus this one, so then I can see what's left, what I haven't taken, the CH3CH2O, and that's what's going to connect up to the carbon here. So let's draw that product. Remember, we keep everything else the same. And we're going to add the connection to the OCH2CH3. So I kind of had to flip that around to get it to work over here because I know that the O is what's going to connect to that carbon. So that O has to end up connecting to the carbon, and then the rest of it connects out from the oxygen. All right, for the next one, same idea. We're going to take the OH from the carboxylic acid, which then becomes water with one of the hydrogens. One of the hydrogens from the amine. And then we're connecting the nitrogen up to that carbon over here. So we've got starting with our carboxylic acid like that. The nitrogen is going to connect here. We remember that we only lost 1 hydrogen to the H2O. Sorry, so let's be careful here and let's write H2O. We only lost 1 hydrogen to the H2O, so that means that the other hydrogen is still there. And then there's going to be a CH3 coming off here. Now we can just write CH3, or we can actually add another line here. Let's do it that way. We could have done that up here too. Remember, line structures, condensed structures, these things are all interchangeable. So you can do it like this. You can do it like this. We could have written CH3 here. We could actually have the line and still write CH3 there. Also okay. I think it looks nice like this, so I'm just going to leave it like that. And then we're going to add that water. And let me know if you have any questions. No, it doesn't have to. The H can go right next to the nitrogen also. It doesn't matter, as long as it's on the nitrogen somehow. Because remember, nitrogen, remember when we abbreviate like line structures, only carbons can lose their hydrogens, meaning that they're not shown. Only carbon is predictable enough that we can not show hydrogens and assume there's a certain number based on how many bonds. Nitrogen, we can't do that. So if we left this hydrogen off, that would be a mistake because that would be actually showing a different kind of a molecule. So whether you put it on the right or the bottom or the left or whatever, doesn't really matter, but it does need to be there. Just like the H on an OH in a line structure needs to be there. All right, any other questions about drawing those reactions? All right. So now, if this is making sense, kind of, let's try something even a little trickier. So one thing that can happen is these reactions can happen intramolecularly, which means they can happen in the same molecule, not just in between 2 molecules. Let me show you what that might look like. So we can take a molecule like this, and if you heat this up with a little acid, there's no other molecule around, but this can actually react with itself. So we can take this carboxylic acid and this alcohol and think about what happens when we lose water there. What's going to happen is now this oxygen over on the left is going to connect to this carbon over on the right. And that's actually, if we count our carbons here, we've got 1, 2, 3, 4, 5. And then the oxygen would be kind of like the sixth member of that ring. So that product would look something like this. Still an ester. Everything's still happening the same way. The difference now is that it's happening within the same molecule instead of in separate molecules. So if we count those again, that's carbon 1, 2, 3, 4, and 5, and then the oxygen. So the oxygen on carbon 5 is now connected to the, carbon number 1, the carbonyl carbon. Just as with these other examples above, it's just happening in the same molecule. And this is called a lactone, and that's important in some flavors and smells and things like that. We'll also see this type of behavior in carbohydrates next week. All right. So the other type of reaction we're going to look at is hydrolysis. And there are actually several different types of hydrolysis reactions. They all involve adding water and breaking something apart. We're going to see hydrolysis in carbohydrates next week. That's a little bit different, but we're going to focus right now on this specific hydrolysis of esters and amides because that is directly related to what we just did. So hydrolysis of esters and amides is really just the backwards reaction of what we just did. So you would take an ester, let's say, or an amide. You add water, and again, this is a reaction that usually needs some sort of a catalyst. So this can be done in acid or base conditions, but there's usually some other thing there. So like H3O plus or NaOH, some kind of water solution that's also acidic or basic. And what's happening then is you're breaking apart the ester at the CO bond and then capping it off with water, water being HOH. So OH goes to one side, H to the other side, and you're essentially doing the opposite of what we just did. So the products are a carboxylic acid and an alcohol. So starting with an ester, you hydrolyze the ester, and you end up with the carboxylic acid and the alcohol. And you can see if you compare that to what we drew up here, it really is the exact opposite reaction. So instead of putting this thing together and having a water byproduct, we're adding water to it, and we're going backwards and forming the carboxylic acid and the alcohol. So we can do the same thing with an amide. Give that a try. See if you can draw those products before I do. In mastering chemistry and the quiz and stuff, it is very specific about what you need to draw here. So it will say, draw the hydrolysis product or draw the condensation product or something like that. So you don't always have a specific reagent here, but you have some more directions. And I believe it'll be that way on the final as well, we'll say draw or it won't be draw because it's a standardized exam, but it'll say which is the hydrolysis product or something like that. All right, you get a chance to draw that. So we're going to again break that CN bond to form the acid. And then our other product here is going to be the amine, like that. Sometimes you'll see this drawn the other way because some people don't like the way that looks, and so sometimes they'll draw like H2N, so it's very clear that the N is connecting to the R, but either way is fine. Okay, let's try some hydrolysis. How do you know where to break it? Good question. So you're always going to break the CO bond of the ester or the CN bond of the amide. Right there. So that's the bond that would form in the condensation direction. So that's the bond that breaks in the hydrolysis. Yeah, so when adding H2O, you're adding two H's and an O. So like in this case, the H2O, so let's say here's our H2O on this bottom one. So the OH is here, and then the other H is here. Up here on the top one, the OH goes to the acid and the H to the alcohol. So that's how we're adding the water. Yep. So another thing I should note, if you go look this up somewhere, like, you're looking up hydrolysis reactions or condensation reactions or whatever, you might encounter something called a mechanism or a curved arrow mechanism, which is a way of accounting for and describing what exactly the electrons do step by step to make these reactions happen. And it's a really good way of understanding the way these things work in general, and organic chemists use it all the time to predict reactions, but it's a little bit beyond the scope of this class. So if you see that and you're seeing these like little arrows being drawn with electrons moving around and different steps and things, that's fine if that makes sense to you, but don't worry if that is confusing because we haven't gone into that. We haven't talked about the details of all that. We're just focusing on if you start with this molecule, what do you get? Or if you start with these other molecules, what do you get as a product? All right, so just as we did before, let's look at some examples of drawing hydrolysis reactions for some larger molecules. All right, so let's look at hydrolysis of this one, see if you can draw that hydrolysis product. And then I'll draw another one as well. And I'm just going to be clear that these are both supposed to be hydrolysis. So a couple challenges here. Go ahead and give them a try while I'm talking. This one, obviously a bigger molecule, so you kind of have to think about what's breaking and what are those products going to look like. This one is actually two amino acids hooked together by a peptide bond. So we're thinking about how we would hydrolyze those amino acids, break them apart. All right. Did you get a chance to try those? So my decision process going through these when I look at ester hydrolysis is the same as with the more written out version like this. I'm looking for the functional group that I know is undergoing hydrolysis, and then where I'm splitting it and capping it just as before. So in this case, if I know there's going to be hydrolysis, I'm looking for my ester or my amide. In this case, the top one's an ester, the bottom one's the amide. So that tells me I know that I'm going to be breaking at that CO bond of the ester, the single bond there. And then once I know that, I can actually just draw those pieces just as a way to organize myself. So if I break that bond, I have this, and I have this. And I don't need to rotate anything or make it, I literally just break it, separate them. Then I can look and say, okay, I know that I need to add OH and H because it's hydrolysis. So OH to one side, H to the other side. And the OH goes with the carbonyl to make a carboxylic acid, and the H goes with the oxygen to make an alcohol. So you can see how I just split those apart and then added the other pieces on there. For this one, this is, so looking ahead a little bit to our protein chapter, this is two amino acids hooked together. This is alanine and glycine. So sometimes that's written like this to show that they're two amino acids hooked together. And they're hooked together by this peptide bond. But again, I don't want to get distracted by all the other stuff going on in this molecule. I know I have to look at, I know I'm thinking about hydrolysis. So I'm looking for esters and amides. And I see here an amide right in the middle. So I know that's where my hydrolysis is going to take place. So that's where I'm going to be end up splitting and then adding my OH and H onto there. Everything else in the molecule stays the same, even though there's these other functional groups there. I don't really need to worry about them. I'm focused on specifically what is undergoing this particular reaction. So that is my hydrolysis. I've got my acid regained and my amine over here. The fact that they're also connected to other stuff, I'm not going to get distracted by that. I'm just drawing my hydrolysis. So this molecule is alanine, and this molecule is glycine. Those are amino acids. And so this is how they might be hooked together in a peptide and then hydrolyzed apart again. There are also other ways of breaking up amino acids, such as through protease enzymes, and we'll get into some of that later. Yeah, hydrolysis, you're always going to break them into two unless, it's some giant thing that has the multiple, that you can break multiple things up. But if you've got an amide, that's going to break into two molecules. You got an ester, that's going to break into two molecules. Yeah, that's how it goes. Other questions about that? All right. So for the final big test of hydrolysis, again, looking ahead a little bit, another molecule that undergoes another realm of molecules that undergo hydrolysis and condensation are the lipids. So as sort of a challenge, we can look at hydrolysis of triglycerides. So a triglyceride might look something like this. Don't be too scared. It's kind of a big molecule here. And it's going to have these fatty acid chains connected to it that are really long molecules. So this would be an example of a triglyceride. And this can undergo hydrolysis to form glycerol. That's where that glyceride name comes from, and some fatty acids. So I'll leave that as a challenge for you to see if you can hydrolyze that thing. In this case, we actually are doing it 3 times. So there are 3 water molecules. You're going to hydrolyze 3 different bonds here to make 4 total product molecules. All right, are there any other questions before we end? For today? All right, well, hopefully that helps. There is still the quiz. I know some of you are all done for the week, so you can have a nice open weekend. We've got 2 more weeks left. We'll do another virtual lab and talk about carbohydrates, proteins, and lipids, and we're done. So we're getting close. So I hope you all have a nice holiday, even though we can't go anywhere. Yeah, don't go in people's houses, and just relax and, I don't know, make some food or something. Let me know if you have questions or if you have issues with the material. I should be able to respond to you this weekend if you need anything. Have a good weekend, everybody. Bye.